Thanks so much. So, try not to speak too loud. The idea was to have a little focus on what we have been developing in bispecific antibodies. We've been speaking a lot about ADCs, but just thinking about having the naked antibody, having a specific action beyond what we are doing today with checkpoints in general. So knocking on two doors, bispecific antibody modulating immune checkpoint blockade. This is my disclosure information. So first of all, this is a table we usually try to complete with the new strategies which are in trials or approved. It's of course a selected panel, but the idea is to see the landscape of what we develop in immunotherapy and trying to understand the positioning of each of the market and how much we can be fast and active in doing that. And as you've seen, the bispecific antibody is probably one of the widest tool being developed today with more or less success through hematological malignancies to solid tumors. And that's a long list, which is absolutely not complete, but is growing over time. Of course, if you think about what we have been proving in solid tumors specifically, is the importance of blocking the PD-1, PD-L1 pathway. So whatever we do, and most of the trials in lung cancer at least, but mainly in solid tumors, keeps this backbone of PD-1, PD-L1 blockade, which is, of course, the paradigm of many monoclonal antibodies or antibodies going only on one target. But it's also the moiety of most of these bispecific today, but not all, as we will see later on. It's important because we had no sessions about immunotherapies that every of these bispecific antibodies that we develop has still to take into account the variability of the patient population and their characteristics when you want to use these patients to try to show superiority as compared to what we had before. So keep in mind that all of these factors might represent important variables that might change your bispecific antibody efficacy, but also might somehow bias or make your trial quite large in order to make sure that replacing an immune strategy by another might be shown superior, which is a challenge now because we have really raised the bar with pembrolizumab and other anti-PD-1, anti-PD-L1, so we have to keep in mind that many factors need to be taken into account if we really want to prove that we go a path forward. So this is a slide from Molly from Hong Kong, who tried to describe the multiple ways we try to anticipate being potentials to improve immunotherapy efficacy. So of course, and my topic is in red on that slide for purpose, we have been trying to combine two checkpoint blockades or two immune checkpoint stimulation or blockades together, including the VEGF if you consider it as a checkpoint, by using two agents. On the left, Empower 150, Orion 31, and others, Ipi/Nivo, tremelimumab, durvalumab. So this is on the left side. What we try to do is to have on a bispecific this dual T-cell function enhancer or T-cell function enhancer combined with the tumor microenvironment remodeling on the same molecule, right? Of course, it might pose some problems of dose adaptation in case of side effects, but the idea is to have on the same molecule the same two functions. Then, of course, you have the T-cell engineering. It was discussed before. You can try to improve the tumor immunogenicity, chemo, ADCs, radiation. But here, there's another type of bispecific, is the dual tumor-associated antigen bispecific. I will come back to there later on. But when you don't target a checkpoint, you have bispecific, which are just here to try to enhance the immunogenicity of the tumor without handling really the immune system. I missed the early part of the previous talk because I was taking my mic, so I don't know, I will be very fast in the introduction. But what is important is in this competitive world, now more than I looked at the latest number, than 350 bispecific antibodies are in development in solid tumors today. Quoted as they can quote it, quantity that they can be counted for. And of course, if you think about overlapping mechanism of action, even if you have two similar targets, right, for example, VEGF and PD-1 or PD-L1, then there are many, you can see on the list on the right, variables for these specific molecules which might make that one of these bispecific antibody works and the other one does not, being the affinities, the valencies, the distance between the two binding sites, the flexibility of the molecule, the real structure of the molecule. So it doesn't mean that they are all equal, even if they have the same targets, okay? This is a wonderful review by Klein and colleagues, just trying to explain how bispecific antibodies can be variable, so you can target two checkpoint inhibitors, So two inhibitory checkpoint pathways, one inhibitory, one stimulatory. You can also put on the other side cytokine modulation, so tumor microenvironment modifiers. You can also have two tumor-associated antibodies, and that's what I called more something which is not handling the immune system, but is increasing the immunogenicity. And as we have been seeing, you have the T-cell engager and the new chapter of the NK cell engager, which are mainly developed today in urothelial cancer and early in breast cancer, a bit less in lung cancer. What is quite important is the slide that was done by Janice, is that we usually in bispecific antibodies follow the principle of combinatorial bispecific antibodies, meaning that you hope that one of the moiety of the antibody will bind, the second, or maybe both would be more specific, but you don't need both to bind to get to your activity, apart, of course, when one of the moiety is a mechanism of action, meaning that if you bridge with T cells, CD3, or NK cells, CD56. So if really one of the moiety is mandatory, obligatory, we call it obligate bispecific antibody, it's specifically the T-cell engager. It will never work without. But you have all of this spectrum of structures. So let me start with one thing which remains very limited, and we'll tell you why. It's a nice story. If you try to target two tumor-associated antigens, so typically amivantamab, you just count on two things, maybe modulating pathways, PI3 kinase, MAP kinase pathway, down your binding, or maybe hoping for an ADCC, antibody-directed cell cytotoxicity, just due to the Fc component of your antibody. So you're not very strong in trying to compete with pembrolizumab, if you allow me to say. So that's the list that we have. We have the amivantamab, which is well established, but not really, as you know, as an immune modulator, but more as a double and dual targeted therapy. And I just would like you to see how we have been moving, because, for example, the izalontamab was a bispecific EGFR HER3. It was developed in nasopharyngeal carcinoma and urothelial carcinoma. And it's still not really passing the shoulder, because this ADCC plus potentially blockade of the EGFR and the HER3 signaling pathway do not really result in strong efficacy. Reason why, it now has been coupled with a topoisomerase I inhibitor, and you have the azolantamab being an ADC. And that's exactly what we did, if you think about this double TA targeting, by developing nowadays ISABRAN. But still, if you think about some example where it worked, of course, you have the amivantamab. I have put the Papillon trial, the Chrysalis and the Papillon trial, but you can put any of them. But on the left-hand side, the Merus component is called pembrolizumab. It's EGFR and LG5, LGR5. LGR5 is a marker of stem cellness, and when you block it, you just block this development of new clones of cell from the very basal and stem cells in the tumor. So it's just blocking the development of the tumor, if I make it short. So this bispecific was shown to have some activity as monotherapy and a very nice activity, so an ASCO this year combined with pembrolizumab in head and neck carcinoma, late line, 67% response rate. So it can work. It's two tumor-associated antigen. You block both pathway, EGFR and the stem cell NES pathway, and then it works very well. But by the way, is it really valid beyond amivantamab in lung cancer? Not really. And when I was telling you the story of maybe transforming this double TAA into ADCs might be better, it's of course the story of putting some topo-1 isomerase on the bispecific HER3 EGFR, and suddenly you have activities in EGFR wild type, in EGFR mutated, and across all oncogene-addicted non-small cell lung cancer, while without the payload, it has no activity at all. So double tumor antigen targeting remains very weak. So of course, what we'd like to target is two checkpoints inhibitory, one stimulatory, or the microenvironment. So of course, you know the long list of checkpoints on the scrutinity, no need to tell them, and this is a current list of what is most promising in terms of trying to have dual checkpoint blockades by using bispecifics. So you can see on this list the CTLA-4 component with phase 3 trials ongoing, the TIGIT component with phase 3 trials ongoing, the LAG3 component, which has been now abandoned, and more the 4-1BB component. Remember, it is a stimulation of the immune system, the CD137, which cannot be given as monotherapy because the activation of the immune system is too strong and gives rise to hepatitis which is not tolerable. So now it's coupled with a checkpoint in order to be able to measure the level of 4-1BB activation by the binding of the antibody. So this is a heat map. I try to, as much as I can again, to put it on the best level in terms of what is being quoted in phase 2 and 3 on clinicaltrial.gov. Of course, we missed the phase 1. So this is the heat map of PD-1, PD-L1 specifically, hitting other targets. So most bispecific have a PD-1, PD-L1 moiety, as I said before, and there are more than 20 VEGF bispecific in development with PD-1 mainly, but also the PD-L1 moiety. And of course, as I said, many, probably lots of phase 1 trials are missed because they are not on clinicaltrial.gov. So this is the most important mechanism. The other mechanism is, again, this 4-1BB, a very potent immune stimulator, which is combined with PD-L1 in many compounds, but is also combined with HER2, is also combined with LAG3, is also combined with HER3 in other bispecific antibodies, but they are in phase 1 and not developed in lung cancer. Okay, so if you think about the interesting double checkpoint inhibition of PD-1, PD-L1, and CTLA-4, that is the list of compounds which today are in the context of large trial being evaluated, mainly in frontline, sometime in later line, the most known for all of us being volrustomig, developed by AstraZeneca. We have been seeing at the ESMO already 2 years ago the nice paradigm-changing trial by Ming-Yu An of this volrustomig plus chemo versus pembro and chemo, which was showing at a high dose of volrustomig the benefits of volrustomig versus the pembro version of the combination. But what we forgot to look at that time was a very small sample size, 20 patients and 21 patients, and what we forgot to stress was the very high toxicity. And later down the road, there was a kind of a proof of concept by using a lower dose of volrustomig of 750, but also increasing this sample size in non-squamous and in squamous non-small cell lung cancer of chemo combined with volrustomig, with, I must say, absolutely amazing response rate, 50 to 60%, a durability and a kind of a paradigm of population response durable over time that really settles the idea of having potentially a higher toxicity than CTLA-4 and PD-1 being blocked separately. So this is in ongoing trials across mesothelioma and non-small cell lung cancer in phase 3 to prove the context, and as you have guessed, with an emphasis for non-small cell lung cancer on low or negative PD-L1, as it was shown before. The LAG3-based cancer immunotherapeutics is still in development. You know, we still are testing in phase 3 a LAG3 inhibitor combined with nivolumab, the same we use in melanoma, the relatlimab. And you remember what is quite interesting with LAG3 is the LAG3 dimer is competing with the T-cell receptor to bind the MHC class 2, mainly in T4, meaning T4 helper cells, right? So what I mean by that, it's a pure competition in the presentation of antigen. So it's absolutely massively logical that it would work to inhibit LAG3, and a bispecific might be an interesting way to really prevent any kind of T-cell activation, no presentation, and exhaustion of the cell. And there was this phase 2 of this bispecific PD-1 LAG3, tobestomig, made sense, with chemo versus pembro and chemo, which unfortunately didn't give any signal of activity. So what is logical in the lab might not be logical in the patients, with the primary endpoint of PFS not being reached, but not any parameter showing any signal of specific subpopulation activity. So very logical, but not proving in the patient. Then I know it's going to be discussed, so I'll go very fast on the VEGF, but I think these names in red, you need to know them because they are reaching the global level. Of course, the ivonescimab for Summit Therapeutics, but the pumitamig from BioNTech, now BMS, combined together, working together on that. The Sunshine Therapeutics bispecific, which now is being developed by Pfizer, and the Lanova bispecific, which now is being developed by Merck MSD. So all of these are now massive phase 3 global trials that, of course, bring to us these strategies in various nuances of the clinical trials. So here I like always to go through some jokes or statistics. I'm sure that, Delvis will show the details, but it's interesting to see that sometimes when a drug is developed in an environment when the requirements are not the same, then the results do not match our requirements. So when you think about the Harmony 2 trial, which was in positive PD-L1 ivonescimab versus pembrolizumab, this strategy was developed mainly for regulators in China, and they made it, right? Wonderful benefit in PFS, and we had this press release, which, by the way, affected a bit the stock option of Summit Therapeutics at the time, but a press release telling that survival was not positive. Okay, but for the survival, instead of balancing the usual PFS and OS alpha, 2.5% of the alpha for each of them, or sometimes even declining a little bit the PFS distribution of the alpha, the 5% you have to distribute. Sometimes doing a strategy of hierarchy where you can recycle your alpha. This trial was giving 0.001% of the alpha to the overall survival, meaning that the significance at less than 0.00001, which was strictly not possible. We did the calculation. I don't know if Summit did it, but with our statistician, we joked to look a little bit what was the hazard ratio, which would have made survival significant. So it was under a hazard ratio of 0.3 for survival to be significant. So that's clearly not adopted to prove an overall survival, but still, you will agree with me, a hazard ratio of 0.77 for survival is something promising, so we probably just need to reproduce it. Same thing for the Harmony 6 trial presented at Presidential at ESMO. It was a debate with the ESMO leadership to be able to have an Asian-only trial being presented as Presidential. Same problem with the SACI-T, by the way. But there's no reason to think that ivonescimab would perform differently in an Asian population as compared to a Caucasian population. This is immunotherapy, so the concept is the same, apart from statistics, as I said before. So ivonescimab plus chemo versus tislelizumab plus chemo. Remember, tislelizumab is EMA approved in patients, very interestingly, in this specific squamous population. We still haven't seen the overall survival data, but the PFS has a hazard ratio of 0.6. We will wait for the overall survival. By the way, the distribution of the alpha and the statistics are very good in that trial. It should be fine to read. And most interestingly for all of us, it completely challenges the dogma that we had since 20 years, that giving a VEGF inhibition, so blocking the proliferation and the, I would say, repairing process of the vessel in squamous was a dangerous thing, has been completely, I would say, disrupted by these trials because it's doable to block VEGF, at least in the context of a bispecific. And the Harmony 3 trial, I hope, will bring results. I hope it's going to be this year to prove it in a Caucasian population. However, I'm not so sure it's needed to always continue to do Asian, Caucasian, instead of just having the paradigm of subset of patients proving the same benefit. 20%, 15% would probably be better. This is the drug developed by BMS and BioNTech, just to tell you a nice activity in PD-1 positive treatment naive patients, but also some activity at resistance to checkpoints. So there's really something to exploit here. And again, this is a drug, a VEGF PD-1 acquired by Pfizer, from Sunshine Pharmaceuticals, where you can see an amazing activity in patients with positive PD-L1 again, but across histologies, across level of PD-L1, so really way better than you would have expected from a monotherapy anti-PD-1, anti-PD-L1. These are the main trials, but the other ones, the one for Pfizer, the one from MSD, are being developed and are being disclosed, but still not officially. So these are the trials, the Harmony 7 in high PD-L1 with ivonescimab, with chemo in all comers PD-L1, all histologies, and the Rosetta Long O2 that I have the chance to lead, which is a phase 2-3 trial with chemo across histologies. So TIGIT. So no need to go too much into this table of TIGIT failures. I participated quite a lot in that, I must say, with lots of antibodies being abandoned, but just TIGIT inhibitors. And you have here the list, abandoned and discontinued. Remember that the TIGIT pathway is not an easy pathway. This is a CD226 pathway, when CD226, mainly in T cells, a bit in NK cells, bind to CD155 on the tumor cells, it leads to tumor cell killing. Okay, this is a pathway of tumor cell killing. But this interaction of CD226 and CD155 has many competitors. One is TIGIT, But CD96 and PVRIG are also competitors. So when you block TIGIT, you might not be really able to restore the binding of CD226 and CD155. That might be the problem. So that's maybe the little picture of TIGIT which makes it not satisfactory, but we have the rilvegostomig. Rilvegostomig is an anti-PD-1, anti-TIGIT, and you have all of a presentation at the World Lung last year showing you how much in ex vivo, so in vitro, ex vivo, it looks like rilvegostomig gives rise to a stronger killing of the tumor cells in the microenvironment of the tumor, probably related partly by this Fc-reduced activity not killing the activated T cells. So something which, at least ex vivo, gives the rationale for continuing to develop this one, and not the other one who have failed. And we have some data, the Artemid-01, which is still growing in terms of number of patients. You can see this was done in patients who were naive from checkpoints. In PD-L1, 1 to 49, more than 50%. And you can see that the patient with or without having received chemotherapy present quite amazing PFS, 6 to 20 months, quite amazing response rate, 30 to 50%. So really an interesting activity. You can look at this table. If you look at high PD-L1 or in 1 to 49, in all of the ranges of response rate, you really improve 20% the response rate in 1 to 49 and 20% also the response rate, 10 to 20 in high PD-L1. So this is why there is all this list of phase 3 ongoing with rilvegostomig today, despite the failure of TIGIT. So really a hope that the ex vivo data will translate. There are other TIGIT, PD-1, PD-L1, which are in development. There's also TIGIT LAG3, TIGIT PVRIG. There's also a TIGIT CD96, and now you understand why, because of the full blockage of the CD226 inhibitory pathways, and they are going into phase 2 in gastric cancer, and even the phase 3 for the BMS compound in gastric cancer. Maybe I think it's the last one, 4-1BB again, PD-L1 4-1BB. So the problem I told you that if you block 4-1BB too strongly, then it will give rise to autoimmune diseases, particularly hepatitis. So the idea of this PD-1 4-1BB, it limits a little bit the binding to 4-1BB to a controlled level because of the bispecific antibody, but you cannot block fully the PD-1 pathway. That the reason why is the acasunlimab has to be potentially combined with a blockage of PD-1 pathway just to fully block this pathway. And you can see that in that trial, in patients who already have been exposed to IO and are resistant to IO, they have a nice response rate of 20% by acasunlimab and pembrolizumab in that trial. And that's why they continue in second line versus docetaxel in an ongoing phase 3 trial with a full blockade, therefore, of the PD-1 pathway. So 4-1BB story is still moving a lot and being developed with this drug trying to block PD-L1 and PD-1 and 4-1BB in order to be using this one. And again, obligate bispecific, you have these T-cell engagers. You know that they move for phase 3. The obrixtamig is also now moving to phase 3. So all of these will have this obligate binding of CD3 plus here DLL3, but there will be others later down the road. There are two maybe prospects I'd like to show you. And here I have a bias. I'm in the Scientific Council of Bicycle Therapeutics. So Bicycle Therapeutics does not antibodies, but peptides. Remember that tarlatamab is not an antibody neither. It's also a peptide. It's a BiTE. But this is a same idea. You can construct small peptide flexible structure with low molecular weight with 2 binding sites on each part of the peptide. And this might have the chance to penetrate better the tissue, penetrate better the brain. You can produce it very easily and massively. So in terms of manufacturing, it's quite easy. And it's possible that it doesn't give rise to ADCC and a little bit less of autoimmune and immune-related toxicities. They have been working a lot, Bicycle Therapeutics, on developing ADCs with a payload on the back of the peptide, but they are also building a lot of bispecific antibodies, particularly in the fields that we explored, EGFR, HER3, HER2. So quite an interesting topic. Remember, for example, just to speak about the unmet needs, that the first bispecific antibody approved by the FDA was this bispecific HER2, the biotropic bispecific antibody in biliary tract disease. So there's really a market here trying to see what we haven't met until now. And last but not least, it's not PROTAB, it's PROTAX. So the proteolysis targeting chimera, which has the idea of binding to EGFR, KRAS, ALK, BRAF, but on the other hand, having a binding to E3 ubiquitin ligase. So this will bridge all of this to the ubiquination machinery, and you can see the E3 ligand that can be used, several of these components which are degraders of the protein, which might allow for some degradation of specific targets, important, of course, for the oncogenic pathway. So this is mostly in preclinical development, but 4 of them have reached phase 1 in non-small cell lung cancer, gastric, and breast cancer. So it's quite nice to see that they will come. The idea is really to degrade proteins which are central in the metabolism of the tumor. So let's see what they come, and thank you for your attention.
Thank you so much.